Welcome to the Deep Dive. Today we're digging into Chagas disease. It's a really serious condition, affects millions, but it's strangely sort of neglected on the global stage. Our goal here for you today is to explore some hope coming from nature, actually. The potential for natural products and finding new treatments.
It's definitely devastating. Chagas disease, caused by the Trypanosoma cruzi parasite. We're talking maybe 6, even 8 million people affected each year, mostly in Latin America, and it causes this severe long-term organ damage. The heart is particularly vulnerable. And it's crucial, as you said, that the WHO calls it a neglected tropical disease. That term really highlights the mismatch, doesn't it? Huge impact, but very limited resources fighting it.
So given that reality, what are patients actually facing right now, treatment-wise? The options seem incredibly limited. Just 2 main drugs, benznidazole and nifurtimox.
Yeah.
And they've been around for, over 40 years?
Yeah, 4 decades. And their limitations are, frankly, quite severe. There's significant toxicity. Think about things like painful skin reactions, bad gastrointestinal issues, nerve damage. These aren't minor side effects. They cause a lot of patients to just stop treatment. Discontinuation rates run from 6% up to 40% in some studies. And efficacy, it's really variable. Okay, in the early acute stage, maybe. But for chronic infections, which is the majority of cases, effectiveness plummets. We're talking 15 to 40%, sometimes less. Plus, and this is worrying, we're seeing parasite resistance emerging. So you put all that together. Toxicity, low chronic efficacy, resistance, and the result. An estimated 1%, just 1% of people who could potentially benefit actually get treated.
1%, that's incredibly stark. It really hammers home the need for something new. So where do we look? You mentioned nature. Why are natural products seen as such a beacon of hope here? Is it just about traditional medicine?
Well, traditional knowledge is definitely part of it, leveraging centuries of use in endemic areas. But scientifically, there's more. First, you get this incredible chemical diversity. Just unparalleled. That means potential for new ways to attack the parasite, maybe hitting targets that current drugs miss, getting around resistance. But here's what I think is really exciting. The dual action potential. Many natural compounds don't just aim to kill the parasite, they often also have potent antioxidant and anti-inflammatory effects. And that's vital for Chagas because a lot of the chronic damage, like the heart problems, is driven by the body's own inflammatory response. So it's a strategic shift, you see. Not just fighting the bug, but also helping to heal the host damage, addressing the inflammation.
Right, that makes sense. It's treating the disease and the body's reaction to it. So are there specific examples? What compounds are researchers actually getting excited about?
Yeah.
And how are they overcoming challenges like getting these compounds to work effectively in the body?
Yeah, definitely. There are promising leads. For instance, compounds called sesquiterpene lactones like Goyas and Sal-Li. These have shown really potent activity, even against strains of the parasite that are resistant to benznidazole. They seem to work by targeting the parasite's unique way of managing oxidative stress, something our cells do differently, which is good for reducing side effects. But a really standout example, perhaps better known, is curcumin, from turmeric. Research shows that combining curcumin with benznidazole, it dramatically boosts the effect. You can actually cut the benznidazole dose by half, which is huge for reducing its liver toxicity. And in animal studies of chronic Chagas, this combination also reduced heart inflammation and fibrosis, the scarring.
Reducing the dose of the toxic drug and tackling the heart damage, that sounds incredibly promising.
Exactly. And then there's the delivery challenge. Many natural compounds aren't easily absorbed or stable in the body. That's where things like nanocarrier systems come in. These are like tiny delivery vehicles. They package the compound, protect it, help it dissolve, and can even target it to infected tissues. These nanoformulations have shown, honestly, outstanding results in animal models, working even against drug-resistant parasites. It's a real game changer for making these compounds viable.
Okay, amazing potential in the lab, powerful compounds, smart delivery systems. But you often hear about this translational gap, getting these discoveries from the bench to the bedside into actual clinics and patients. What's needed to bridge that gap for Chagas?
You're right, that gap is real. We still have limited clinical trials for these new approaches, especially natural product-based ones. Bridging it requires several things. Deeper understanding of exactly how these compounds work, their mechanisms, developing what you might call intelligent therapeutic strategies, maybe combining natural products, maybe with lower doses of existing drugs, using these advanced delivery systems. But crucially, it needs sustained effort, collaboration. It's not just the science, it also demands socio-political will. We need commitment to tackle affordability, accessibility, and fundamentally addressing the systemic neglect that has allowed Chagas to fester for so long.
It really paints a picture, doesn't it? from this overlooked disease, tapping into ancient wisdom, and pushing the boundaries with cutting-edge tech like nanomedicine.
Absolutely. And I think this deep dive really underscores a broader point for global health. Fighting diseases like Chagas isn't just about searching for one single magic bullet drug. It's about building a more holistic approach, one that integrates insights from nature, scientific innovation, advanced technology, and, critically, the commitment to tackle the social and economic roots that let these diseases persist. You really have to understand that whole complex picture to find solutions that actually last.
Welcome to the deep dive. This is where we try to crack open complex information, sift through, stacks of sources, and really pull out those crucial, sometimes surprising insights. Today we're plunging into a topic that, well, it might seem quite distant for many, but Chagas disease is actually a truly global health challenge. And despite its pretty significant impact, it often flies under the radar. So our mission today is to explore this scientific race to, well, hopefully conquer Chagas disease. We'll look at the desperate need for new treatments, and especially the cutting-edge research that's leveraging nature's own ingenious solutions. We've got a really fascinating stack of research papers, reports, and we're going to try and extract what's genuinely important and frankly quite remarkable about this often neglected disease.
Yeah. And Chagas disease, or you might hear it called American trypanosomiasis, it's caused by this parasite, Trypanosoma cruzi. The World Health Organization recognizes it as a neglected tropical disease. And while, yes, its roots are in Latin America, its reach now is surprisingly global. I mean, because of increased migration, you find established patient populations in places like the US, Europe, particularly Spain, actually, Australia, and Japan. Globally, we're talking about an estimated 6 to 8 million people affected. And the economic burden, it's huge, over $7 billion annually in healthcare costs and lost productivity.
Wow, 7 billion. For a disease many of our listeners, I bet, might not have even heard of before. And when you look at the current treatments, the need for something new becomes incredibly clear, right?
Absolutely. The standard drugs, benznidazole, we call it BNZ, and nifurtimox or NFX, they were developed way back in the 1970s.
70s, that's a long time ago in medical terms.
Exactly. And the problem is they come with serious drawbacks. High toxicity, for one. They can be really brutal for patients. Then there's a treatment duration, 60 days. Imagine 2 full months taking highly toxic drugs.
60 days, that's a huge commitment.
It is. And on top of that, their effectiveness, their efficacy, it's variable, especially once the disease has become chronic. So yeah, the hunt for better, safer alternatives isn't just important, it's absolutely vital.
So where are researchers focusing that hunt? You mentioned nature's solutions earlier.
That's right. And what's truly fascinating here is why the natural world, especially medicinal plants, has become such a central focus for drug discovery in Chagas. The thinking behind it is really compelling. Plants, they've evolved over millennia, developing these diverse, sophisticated chemical defenses against their own pathogens. This means they produce an incredible array of unique natural compounds. We call them secondary metabolites. And these often have potent biological activities.
Okay, so they're like nature's own little chemical weapons factories.
In a way, yes. And crucially, these natural compounds often have novel chemical structures, different ways of working, mechanisms of action that you just don't find in our synthetic drug libraries. So this offers real potential to maybe overcome parasite resistance or target new pathways within the parasite itself, basically hitting it where it's most vulnerable, using strategies inspired by nature.
It really sounds like a potential gold mine. But I imagine tapping into that, well, it must come with some significant hurdles.
Oh, absolutely. Our sources definitely highlight several major challenges. For starters, there's a real lack of standardization in testing protocols across different labs, different studies, that makes it hard to compare results directly.
Apples and oranges.
Exactly. Plus, you've got the natural variability in the plant extracts themselves. The same plant grown in different conditions might have different levels of active compounds. Then there's the sheer difficulty in isolating and identifying the specific active compound from these complex mixtures. And maybe the biggest hurdle is moving from promising results in a lab dish.
The in vitro stage.
Right, the in vitro screenings. Moving from that to rigorous studies in animal models, the in vivo studies, that actually reflect the human disease. It's a long, complex path.
Okay, so it's not easy. But despite those hurdles, it sounds like some really remarkable discoveries are emerging. Let's dive into some specifics. You mentioned plants, families, compounds.
Yes, let's do that. Take terpenoids, for example. Specifically, a group called sesquiterpene lactones. These are incredibly abundant in the Asteraceae family, think daisies, sunflowers.
Okay.
So two compounds, goyasin solid and lignopholid. They were isolated from Brazilian plants, from the Erymanthus and Lichnophora genera. These have shown really potent activity against all three forms of the T. cruzi parasite in lab studies.
All three forms. That's impressive.
It is. But what's even more remarkable is they've actually demonstrated curative effects in animal models of acute Chagas disease.
Curative. Wow.
Yeah. And their mechanism seems to involve hitting essential parasite enzymes like trypanothione reductase. That's an enzyme unique to the parasite, vital for its internal defense system. It's like a precision strike, really, inspired by the plant's own defenses. And beyond those, there are others, like deoxymecanolide from Mecania micrantha. That also showed significant efficacy, reducing the parasite load in the blood, the parasitemia, in infected mice.
And you mentioned Artemisia earlier, like the source for malaria treatment.
Exactly. That's a great connection. Artemisia species, they produce artemisinin, which revolutionized malaria treatment. Well, various Artemisia species, including Artemisia absinthium, that's common wormwood, had also shown activity against T. cruzi. It really highlights how these anti-parasitic natural products might have broader applications.
That's just one fascinating chemical family, though. The plant kingdom is huge. What else are researchers finding?
Well, another really interesting group is phenolic compounds. These are quite valued because they seem to have this dual action potential.
Dual action. How so?
They can directly kill the parasites, but they also often possess powerful antioxidant properties. And that second function is critical because oxidative stress is a key part of the damage in Chagas heart disease. So these compounds might help mitigate that damage.
Ah, so fighting the bug and protecting the heart tissue.
Precisely. A good example here are naphthoquinones, like lapachol and beta-lapachone. These come from the bark of the Tababuia species. You might know it as lapacho or paudarco tree. These were actually among the very first natural products studied extensively for Chagas. They showed potent activity, but unfortunately, human trials were ultimately stopped due to toxicity issues.
Oh, boy.
It was. But even so, they remain really important lead structures, sort of blueprints for developing potentially less toxic synthetic versions. Then you have something like curcumin. That's the active compound in turmeric, right? Curcumin.
Turmeric, yeah. Known for being anti-inflammatory.
Exactly. It's a powerful anti-inflammatory and antioxidant. Now, while it does have some direct activity against T. cruzi, its more important potential might actually lie in combination therapy. Studies in animal models showed that when curcumin was used alongside benznidazole, the standard drug,
Hey?
it actually helped reduce cardiac inflammation and fibrosis, the scarring in the heart muscle. This strongly suggests it could be a valuable adjuvant therapy, a supporting treatment to protect the heart while the main drug kills the parasite.
That makes a lot of sense, using it to manage the collateral damage.
Exactly. And there are other phenolics too, like flavonoids, quercetin, for instance, which you find in lots of common fruits and vegetables. These can also inhibit key parasite enzymes and help protect host cells from that oxidative damage we mentioned.
So we're really talking about compounds that not only attack the parasite directly, but also bolster the host's defenses or mitigate the damage. That feels like a much smarter approach. What else from the plant kingdom is showing promise?
Another diverse group we should look at is alkaloids. These are nitrogen-containing compounds, a huge variety of them. From the Ananaceae family that includes plants like soursop and pawpaw, we get Ananaceous acetogenins. These have shown really potent activity against T. cruzi.
How do they work?
They seem to inhibit something called mitochondrial complex I. Basically, they shut down a crucial part of the parasite's energy production system, starving it of power, essentially.
Wow. Okay.
And another interesting one is piperine. That's the main alkaloid from black pepper.
From black pepper, really?
Yeah. Now, its direct activity against the parasite is only modest, but its real value might be as what's called a bioenhancer.
A bioenhancer.
Right. Piperine is known to inhibit certain metabolic enzymes in our bodies. So when you take it with another drug, it can improve the absorption and bioavailability of that other drug.
Ah, so it helps the main drug work better or last longer?
Exactly. It means you might potentially need a lower, maybe safer dose of the conventional drug if piperine helps your body utilize it more effectively.
That's a really clever strategy, using one compound to boost another. And I recall our sources mentioned a few other plant extracts being looked at too.
Yes, the scope of the research is pretty wide. Various salvia species, for instance, are being tested. Aristogrecia glutinosa, that's another one. A compound isolated from it showed significant activity in animal models. Even essential oils from plants like Zetaria multiflora have shown some activity, though often in very early lab studies. It just underscores this incredible potential hidden in nature's vast chemical library.
Okay, these plant-based discoveries are definitely exciting. But to really grasp their potential impact, maybe we should zoom out a bit and understand the bigger picture of Chagas disease itself. Because it sounds like a much more complex adversary than many people might realize.
That's absolutely right. I mean, the parasite's transmission is way more complicated than just the kissing bug. That classic vector-borne route, through the feces of infected triatomine insects, it's still very significant. But what's quite surprising and increasingly important are the non-vectorial routes.
Like what?
Well, for instance, congenital transmission, mother to child. This occurs in about 5% of pregnancies in women who are infected. The crucial thing here, though, is that therapy for infected newborns is nearly 100% curative if it's given right at birth, which really highlights how critical early diagnosis is.
That's good news, at least for newborns. What else?
Transmission via blood transfusions and organ transplants still poses a risk, even with screening programs in many places. But here's where it gets really interesting and frankly quite concerning: oral transmission.
Oral. You mean eating or drinking the parasite?
Essentially, yes. Consuming food or drinks contaminated with parasite-laden insect feces, or sometimes even the infected insects themselves accidentally crushed into food preparations. This has emerged as a major cause of acute outbreaks, often with high mortality rates, especially in the Amazon basin.
Wow. Can you give an example?
Sure. There was a well-documented outbreak in 2007 at a school in Caracas, Venezuela, affected over 100 people. And there have been recurrent outbreaks in Brazil linked to things like contaminated artisanal acai fruit pulp and sugarcane juice.
Acai and sugarcane juice. Things people consume regularly. That completely changes the picture from just a bug bite in a rural hut.
It really does. It moves the risk into different environments and populations. It reshapes how we have to think about prevention and control.
And the disease itself, its clinical course, is also quite distinct, isn't it? It has phases.
Yes, it's typically biphasic. You have the initial acute phase, which lasts, weeks to maybe a couple of months after infection. Often this phase is asymptomatic, or people just have mild, nonspecific symptoms, maybe a fever, feeling unwell. So it frequently goes completely undiagnosed.
Which means people don't even know they have it.
Exactly. However, those oral infections we just talked about, they often present with a much more severe acute illness, intense abdominal pain, vomiting, sometimes gastrointestinal bleeding. It's a much more dramatic onset.
Okay, so after the acute phase, what happens if it's not treated?
If it's left untreated, the infection enters the chronic phase. Now, for about 60 to 70% of people, they remain asymptomatic indefinitely. This is called the indeterminate form. They carry the parasite, but don't have obvious symptoms, maybe for their whole lives.
Not everyone is that fortunate.
No. For the remaining 30 to 40%, the disease slowly progresses over decades, eventually leading to symptomatic forms. The most common and, frankly, the most devastating is chronic Chagasic cardiopathy, or CCC.
The heart problems.
Right. This leads to gradual heart enlargement, heart failure, dangerous arrhythmias, electrical conduction problems, and a significantly high risk of stroke and blood clots, pulmonary embolisms, largely because the disease creates this prothrombotic state, making clots more likely to form.
That sounds incredibly serious, developing over years without knowing.
It is. And then there's a less common form, the digestive form. This involves damage to the nerves in the gastrointestinal tract, leading to conditions called megaesophagus and megacolon, basically. The esophagus and colon become massively enlarged and lose function, causing severe problems with swallowing and digestion.
So what's actually driving this damage in the chronic phase? Is it just a parasite hanging around?
It's more complicated than that. The underlying pathology seems to be this complex triad. Yes, parasite persistence plays a role, even if at low levels, but also crucial is a dysregulated host immune response. The body's own defenses kind of go haywire and contribute to the damage. And thirdly, there's microvascular damage problems with the small blood vessels. And interestingly, the specific outcome for any given person seems to be influenced by factors like the genetic strain of the T. cruzi parasite they were infected with, and also the host's own genetic makeup.
Wow, it really is a multi-layered challenge. And I guess that complexity must make research and testing new drugs incredibly difficult too.
Absolutely. You've got that lack of standardization in testing we mentioned earlier. You've got the huge genetic diversity within T. cruzi itself. Different strains respond differently to drugs. Plus, animal models, while useful, don't always perfectly replicate the slow, progressive fibrosis, that scarring we see in human chronic heart disease. So making direct comparisons between studies or predicting human outcomes from animal data is really tough. It definitely hinders rapid drug development.
It sounds like a researcher's nightmare in some ways.
It can be, but despite these difficulties, some really exceptional findings and human studies have given us critical insights.
Like what? What have we learned from studying people?
Well, for example, HIV co-infection. We now know that in people with advanced HIV/AIDS, latent Chagas infection can reactivate, and it's often severe, frequently causing devastating brain lesions. It really changes how we need to think about screening and managing Chagas in immunocompromised groups.
That's a critical interaction.
Definitely. Another thing, remember that Caracas school oral outbreak. A 10-year follow-up study of those individuals revealed really high rates of persistent parasites long after the initial event. It underscored just how severe oral transmission can be and the need for aggressive, possibly prolonged treatment. We've also seen fascinating links between nutritional factors and disease severity. For instance, studies found that lower levels of selenium in the blood, selenium is a crucial antioxidant cofactor, were associated with more advanced forms of Chagastic cardiomyopathy.
Suggesting oxidative stress is a key player in the heart damage.
Exactly, it reinforces that idea. Also, we've confirmed that chronic Chagas induces a prothrombotic state, increasing stroke risk. But crucially, studies have shown that giving anti-trypanosomal treatment like benznidazole can actually partially reverse these clotting abnormalities.
So the treatment can help with that specific complication.
It seems so. And finally, there's growing evidence for host genetic susceptibility. Variations in our own genes, particularly those involved in the immune system like HLA alleles or cytokine genes, seem to influence whether someone stays asymptomatic or progresses to severe heart or digestive disease.
Okay, so bringing all this together, the complexity, the genetics, the different transmission routes, what happens when potential treatments actually get tested in large human trials? What has the reality check been?
That's the million-dollar question, isn't it? The benefit trial was a real landmark here. It was huge, over 2,800 patients who already had established chronic Chagas heart disease. And the result was, well, sobering. It fundamentally reshaped how we think about treating chronic Chagas.
What did it find?
It found that while benznidazole did successfully clear parasites from the blood in many patients, it did not significantly reduce the main outcome they were looking for, which was a combination of clinical progression events like worsening heart failure, stroke, or death compared to the placebo group.
So killing the parasite didn't stop the heart disease from getting worse in those patients.
Not significantly in that specific trial population with established heart disease. Now, this wasn't necessarily a failure of the drug itself, but a crucial insight. It suggests that, at least in the advanced chronic stage, the damage might already be somewhat irreversible. Or perhaps it's driven more by those inflammatory processes that just keep going even after the parasite numbers are reduced.
Right, so just killing the bug isn't enough at that late stage.
It seems not. It really forced researchers to think beyond just being parasiticidal and towards maybe needing combination approaches that also tackle the inflammation or fibrosis. Now, other trials have provided more nuances. The Bendita trial suggested shorter courses of BNZ might be effective for people in the indeterminate chronic phase before major heart damage occurs. And the Stop Chagas trial added to the evidence that antifungal drugs called azoles, while promising in the lab, weren't sufficient on their own for a sustained cure in humans.
And diagnostics are evolving alongside this too, I imagine, moving beyond just looking for the parasite.
Absolutely. We're seeing more advanced techniques like metabolomics, studying the small molecules in blood, and mass spectrometry being used to try and find serum biomarkers. The hope is to find markers that could predict who's going to progress to severe disease. And molecular methods like qPCR, that's real-time quantitative PCR, a very sensitive way to detect and count parasite DNA, are now pretty standard for monitoring how well treatment is working, at least in terms of clearing parasites from the blood.
But is there a definitive test for cure?
That's still a major challenge, actually. There's no universally accepted biomarker of cure. Parasite DNA can sometimes persist in tissues, even when it's undetectable in the blood after treatment. So knowing for sure if someone is truly cured remains difficult. And all of this just brings us back to the urgent need for new, better solutions, especially given those limitations of BNZ and NFX we discussed.
Right, the toxicity, the long duration.
Exactly. And while there are some new related drugs being explored, like fexinidazole and those antifungal azoles we mentioned showed promise in vitro, they've largely failed in clinical trials to achieve a sustained cure. The thinking now is that many azoles might be primarily parasitostatic. They stop the parasite from multiplying rather than truly parasiticidal, meaning they actually kill the parasite outright. And for a persistent infection like Chagas, you probably need something that really eliminates it.
Okay, so if the single magic bullet approach isn't working, especially for chronic disease, what's next? It sounds like the field is shifting towards more sophisticated strategies.
Precisely. That's where the really exciting innovation is happening now. We're recognizing that a multi-pronged attack is probably needed.
Like what kind of approaches?
Well, one key area is synergistic combinations, using multiple drugs or compounds together.
Like the curcumin with benznidazole you mentioned earlier.
Exactly like that. Combining natural products with standard drugs to maybe enhance efficacy or reduce the toxicity of the standard drug or provide complementary benefits like reducing inflammation. We're also seeing combinations of conventional drugs like BNZ plus an azole, which has shown promise in animal models, even if ashills alone weren't curative in humans.
Okay, combinations make sense. What else?
Something truly groundbreaking, I think, is the exploration of immunotherapy combined with BNZ.
Immunotherapy, like harnessing the immune system.
Yes. In experimental models, researchers tried combining a reduced dose of BNZ, potentially making it safer, with a therapeutic vaccine designed to modulate the immune response. And the results were remarkable. This combination didn't just control the parasite, more importantly, it actually seemed to calm down the host's destructive immune reactions, leading to a superior restoration of heart function compared to BNZ alone.
Wow, that really is a paradigm shift. Treating the host's reaction as well as the bug?
It really is. It moves beyond just killing the parasite to actively trying to fix the underlying pathology driven by the immune system. We're also seeing efforts in repurposing drugs. For example, testing drugs like disulfiram, which is actually used to treat alcoholism to see if it can help mitigate the adverse side effects of benznidazole.
And what about getting the drugs to where they need to go more effectively?
Ah, yes, that's another huge area. Nanocarrier delivery systems. This tackles a major problem. Many promising compounds, both natural ones like those sesquiterpening lactones and synthetic ones, have poor water solubility or low bioavailability. They just don't get absorbed well or reach the target tissues effectively.
So nanotechnology can help with that.
Exactly. Researchers have successfully encapsulated compounds like sesquiterpening lactones and even benznidazole itself into tiny nanocarriers. And these nanoformulations have shown really outstanding efficacy in animal models, impressively, sometimes even against parasite strains that were resistant to standard benznidazole.
How do they achieve that?
Well, these nanocarriers can improve the drug's stability, allow for sustained release over time, and potentially even be designed to target the drug more specifically to infected tissues. This could dramatically enhance effectiveness while potentially lowering the overall dose needed, thus reducing toxicity.
That sounds incredibly promising for improving existing drugs and enabling new ones.
It really is. And one final emerging concept is targeting host pathways directly. The idea here is to combine a drug that kills the parasite with a separate agent that specifically targets the host's damaging responses, maybe an anti-inflammatory drug or an anti-fibrotic drug to combat the scarring.
So really tackling both sides of the equation, the parasite and the host's damaging reaction to it.
Precisely. The ultimate goal now seems to be creating safer, more effective combination therapies. Therapies that don't just kill T. cruzi, but also actively protect the host from the devastating chronic inflammation and tissue damage that really defines the long-term impact of Chagas disease.
We've certainly covered a lot of ground today, taking an incredible journey through the complex world of Chagas research, from the frankly inadequate drugs we've relied on for decades, to the astonishing potential being uncovered in herbs and natural products. And then looking forward to these really cutting-edge strategies like nanocarriers, combination therapies, and even immunotherapy. It's clear that while there still is significant translational gap, that gap between promising lab results and treatments actually reaching patients, the exploration happening right now offers a genuinely rich and promising frontier.
Absolutely. And perhaps it leaves us, and you, the listener, with a thought to consider. How does this ongoing discovery, particularly within the natural world, continue to reshape our fundamental understanding of medicine? It really prompts us to look in sometimes unexpected places, like traditional plant knowledge, for solutions to these incredibly complex global health challenges. And it makes you wonder, doesn't it? What other neglected diseases or health problems might have incredible natural solutions?
Hello, we're excited to show you our native speech capabilities, where you can direct a voice, create realistic dialogue, and so much more. Edit these placeholders to get started.
Marco,
Dr. Vance, thank you so much for meeting with me. My research on Chagas keeps leading me back to the same problem: the harsh side effects and limited efficacy of benzonidazole and nifurtimox. It feels like we're stuck.
It's a common frustration, Marco. That's precisely why my field is so active right now. We're not just looking for a replacement. We're looking for a smarter, more holistic approach. And for that, we often turn to nature, which has been conducting its own chemical warfare experiments for millions of years. Where would you like to start?
Let's start with the heavy hitters. I've read a lot about the Asteraceae family, the daisy and sunflower family, being a hotbed of activity. Can you elaborate on that?
An excellent place to begin. The Asteraceae family is famous for producing a class of compounds called sesquiterpene lactones. Think of them as the plant's highly specialized chemical weapons.
And which plants are the most promising?
In Brazil, a lot of focus is on species of Lichnophora and Eremanthus, which are often referred to locally as Brazilian arnica. From these, we've isolated compounds like lignopholide and goiazenzolide. Their effect on Trypanosoma cruzi is profound. They are highly parasiticidal, meaning they directly kill the parasite. Their mechanism is aggressive. They are alkylating agents, which means they can bind and deactivate essential parasite proteins, particularly the enzyme trypanothionine reductase. This enzyme is unique to the parasite and is vital for its defense against oxidative stress. By knocking it out, the parasite is left defenseless and dies. In animal models, these compounds have even led to sterile cure in the acute phase.
That's incredible. And what about its other health benefits? Why is it called Brazilian arnica?
That's the ethnobotanical link. Traditionally, these plants are used topically, just like European arnica, Arnica montana, as a potent anti-inflammatory for bruises, sprains, and wound healing. So this compound family has a dual legacy: a traditional, observable anti-inflammatory effect, and a scientifically validated, potent anti-parasitic effect. It's a perfect example of how traditional knowledge can point us toward powerful biochemistry.
You mentioned oxidative stress. My understanding is that much of the heart damage in chronic Chagas isn't from the parasite itself, but from the body's own out-of-control inflammatory and oxidative response. Are there any herbs that specifically target that?
Exactly. And that's where we move from direct killers to strategic allies. The superstar in this category is turmeric, or curcuma longa.
I think of curcumin for arthritis and general inflammation. How does it fit into Chagas treatment?
Its direct effect on T. cruzi is modest. However, its true power lies in being an adjuvant therapy. The chronic inflammation in chasic cardiomyopathy leads to fibrosis, the scarring that stiffens the heart muscle and leads to failure. Curcumin is a master anti-inflammatory and antioxidant. In animal studies, when curcumin was given alongside benzonidazole, the benzonidazole killed the parasites, but the curcumin significantly reduced the cardiac inflammation, oxidative damage, and fibrosis. The mice treated with the combination had much healthier hearts than those treated with benzonidazole alone.
So it's not a replacement, but a partner drug. It treats the host's pathology, while a conventional drug treats the infection.
Exactly. This is the future of Chagas therapy. As for its other uses, the list is endless. In Ayurvedic and traditional Chinese medicine, it's used for everything from digestive disorders and liver problems to skin diseases and depression. Modern medicine is actively studying it for Alzheimer's, cancer, and metabolic syndrome. It's a true polypharmacological agent.
What about other plants that were once promising? I've seen papers on lapacho or pau d'arco.
Lapacho, or Tabebuia impetuogenesis, a classic and a cautionary tale. Its active compounds, lapichol and beta-lapichone, are a type of naphthoquinone. They are exceptionally potent against T. cruzi in vitro. They work by generating reactive oxygen species, essentially overloading the parasite with oxidative stress. Traditionally, Lapacho tea is a panacea in South American folk medicine, used for infections, fevers, and even cancer. The problem? The dose required for a therapeutic effect in humans proved to be too toxic, causing severe internal bleeding. It highlights the critical challenge in natural product research, finding the therapeutic window between efficacy and toxicity.
That makes sense. Let's shift to a different class of compounds. I've read about alkaloids being investigated, particularly from plants like soursop.
Yes, the soursop tree, or Annona muricata, and its relatives produce a unique class of compounds called antinoceous acetogens. These are incredibly potent. Their mechanism against T. cruzi is to inhibit complex 1 of the mitochondrial electron transport chain. In simple terms, they cut off the parasite's main energy supply at the source. It's a very effective way to kill it.
And its other uses? I've seen it promoted heavily online.
That's another complex story. Traditionally, the leaves are used for teas to treat fevers and calm nerves. In the world of alternative medicine, it has gained immense popularity as a cancer treatment, though robust clinical evidence in humans lacks. There's also a significant concern. High consumption of these plants has been linked to atypical Parkinsonian syndromes in the Caribbean due to potential neurotoxicity. So, like Lapacho, it's a powerful plant that demands extreme caution.
It seems like the most promising strategies involve combinations. Are there any helper herbs that might make other compounds work better?
A brilliant question. You're thinking like a pharmacologist now. The answer is a resounding yes, and the best example is black pepper, or Piper nigrum.
I thought that just made things spicy.
It does, but it's also a powerful bioenhancer. Piperine inhibits certain metabolic enzymes in our liver and intestinal wall, particularly CYP450 enzymes, which are responsible for breaking down drugs and other compounds. By inhibiting them, piperine allows other active compounds, like curcumin, for example, to stay in the bloodstream longer and at higher concentrations. Its direct anti-trypanosomal effect is weak, but if you formulate a treatment of, say, benzonidazole and curcumin, adding piperine could make the whole formula more potent and bioavailable.
So the ideal treatment might be a cocktail: benzonidazole to kill the parasite, curcumin to protect the heart, and piperine to make sure the curcumin works effectively.
You've got it. That's the essence of modern ethnobotany and drug development. It's not about finding a single magic bullet. It's about building a multi-pronged, synergistic strategy. We use direct-acting compounds like sesquiterpene lactones, protective adjuvants like curcumin, and bioenhancers like piperine. We can even package these compounds into nanocarriers to deliver them more effectively. The goal is to create a therapy that is lethal to the parasite, gentle on the host, and addresses both the infection and its devastating consequences. It's a complex puzzle, but every plant we study gives us another potential.
Welcome to the Deep Dive. We unpack complex topics, pulling out the essential insights for you. Today, we're tackling a global health challenge that honestly still flies under the radar for many: Chagas disease. It's this persistent parasitic infection, right? Also called American trypanosomiasis, caused by a tiny parasite, Trypanosoma cruzi. Now, it hits millions, especially across Latin America, but here's the thing. We're actually seeing it pop up, even local outbreaks, in places you wouldn't expect, like the southern U.S., parts of Europe. It really highlights that this is becoming more of a global concern now. And the way this disease works is, well, it's tricky, often silent. It kicks off with an acute phase, could be mild, maybe no symptoms at all. But then for maybe 40% of people infected, it quietly morphs into what they call determinate chronic Chagas disease. And that's when the really serious, life-changing problems start. We're talking major heart and digestive issues, sometimes even decades later.
Yeah, what's really insidious, I think, is how it just simmers under the surface for so long. The most common, and frankly the most severe outcome, is Chagas cardiomyopathy, CCM. That affects nearly a third of people with chronic Chagas. And the prognosis there, it's poor. We're looking at maybe a 4% mortality rate each year. Just imagine that. A disease quietly damaging your heart, relentlessly, and you might not even know until things are pretty advanced. That's the reality for so many.
And getting a diagnosis, especially for the gut problems, that sounds like a real challenge too. I mean, doctors really have to dig deep sometimes, right? What kind of tests are we talking about?
Exactly. For that gastrointestinal involvement, they often use imaging studies, things like barium swallows, manometry, endoscopy, CT scans, x-rays. These aren't standard for everyone. Maybe about 1 in 10 patients with GI issues need them. But they're crucial. They can show up really specific, severe problems like megaesophagus or megacolon, basically extreme enlargements of the esophagus or the colon. And that, as you can imagine, severely messes with eating, digestion, leads to malnutrition, all sorts of complications.
Okay, so diagnosis is tough. What about treatment? The current picture is, well, it's pretty limited, isn't it? I gather it's curable if you catch it super early, in that acute phase. But the drugs we have now really struggle once it's become chronic. And that's exactly why we're doing this deep dive today. Our mission is to unpack the latest research, looking at new compounds, plant extracts, fascinating findings from human studies, all trying to find better ways to help people living with Chagas.
Right. And if we sort of zoom out a bit to the bigger picture of finding these new treatments, a lot of the studies are focusing strategically on specific targets within the parasite itself, enzymes it needs to survive. Think of them like the parasite's Achilles heel, things it absolutely needs, but which are maybe unique to it or very different from us. Two big ones are trypanothione reductase, that's crucial for the parasite's defense against stress, and it's unique to these types of parasites, not found in humans, makes it a great target. And the other is squalene synthase. That one's key for building its cell membrane, basically its outer wall. So hitting these is like taking apart the parasite's essential toolkit.
Wow, okay. So it sounds like the research net is being cast really wide. Given how tough the current drugs are, is this sort of a hedging bets strategy, exploring so many different types of compounds, or is there a clear logic?
Oh, it's definitely strategic diversity. You don't want to put all your eggs in one basket, right? Especially with a complex parasite like T. cruzi and the known issues with current treatments. Pursuing multiple avenues, synthetics, natural products, different targets, it just increases the odds of finding something truly effective and hopefully safer.
So let's talk about the synthetic side first. How are they building these new potential drugs in the lab? Sounds like a couple of interesting approaches. One involves metals, organometallic shift bases.
Yes, exactly. Those are derived from compounds like 5-nitrothiophene and 5-nitrofuran. The idea is that incorporating a metal atom might give the molecule unique ways to interfere with the parasite's own metal-dependent processes. It's a targeted approach. And then there's another angle, focusing on nitrogen-rich structures, like these novel 3-nitro-1-H1 really 2-gal-triazole-based amides and sulfonamides. These kinds of structures are often good at disrupting metabolic pathways.
Interesting. And you also mentioned isoxazole analogs, compounds 12A, 12B, 12C,
Right.
synthesized using microwaves, which sounds pretty cutting edge, speeding things up, and based on natural plant compounds called lignans.
That's right. They drew inspiration from natural structures like Varigensin and grandison. Using microwave irradiation can, yes, significantly accelerate the chemical synthesis process. What was really notable about these specific compounds was their potency against the amastigote forms of T. crus. Those are the forms hiding inside our own cells, which are notoriously difficult to reach. But interestingly, they didn't work against the trypomastigotes, the forms circulating in the blood.
Huh. So different drugs might target different stages. That's important.
Absolutely. And this leads us nicely to the natural side of things. What about plants? We're seeing a lot of research digging into plant extracts, often building on traditional uses but applying modern science. For example, studies on bee uncinella using ethanolic extracts have pinpointed several active compounds, things like caffeic acid, pectolinaringenin, hispidulin, and even a mix of 3 chrogenic acids. And the exciting part, while they showed activity against T. cruzi, they also seem to have low toxicity towards our own cells, like red blood cells and immune cells, macrophages.
That safety profile is key, isn't it? Especially for a chronic disease.
It's absolutely critical. You need something people can take long-term without devastating side effects. And just to broaden that point slightly, The whole plant genus Acalypha, which B. encinella belongs to, is seen as a promising source for antiprotozoal agents in general. It shows this wider trend, looking to nature's pharmacy for new leads.
Okay, so that's the research pipeline. But what does this mean for someone actually living with Chagas today? Because the reality is those frontline drugs, benzimidazole and nifurtimox, they come with real challenges. Imagine having to take a drug for months, maybe years, knowing it could cause really bad nausea, skin reactions, nerve issues. That's what patients face. It makes sticking to the treatment incredibly difficult, especially for adults. I read that side effects are so common, something like 15–20% of patients just stop taking the medication. And it tends to hit adults harder than kids. It really drives home why we need safer options, urgently.
You've hit the nail on the head, and that's why some research is looking beyond traditional drugs, too. There was one fascinating study, a randomized controlled trial. They screened 85 patients, ended up randomizing 66. Half got selenium, the other half got a placebo. They were looking at it as a potential non-traditional intervention for chronic Chagas.
Selenium, like the supplement.
Exactly. Exploring if something like that could offer supportive benefits. The fact they're even testing this shows the need for alternatives. And importantly, in that specific trial, the few patients who did stop treatment didn't report adverse effects related to the selenium. It's this kind of human trial data, even for supportive therapies, that gives us really crucial real-world insights.
Right, and thinking about the body's own defenses, what happens to the immune system after someone gets treated? Are there noticeable changes?
That's a really important area of study, and yes, there are some interesting findings. Researchers looked at specific immune cells, CD4 plus T cells, which are key orchestrators of the immune response. What they saw after treatment was a significant decrease in certain T cells that express multiple inhibitory receptors, basically brake signals on the immune system. Things like 2b4 plus CD160 plus PD1 plus SMI, or 2b4 plus CTLA4 plus PD1 plus TCAT. These cells normally kind of quiet things down. Seeing fewer of them suggests the treatment might be helping to release the brakes on the immune system.
Letting the body fight back more effectively?
Potentially, yes. And these changes weren't just temporary. They saw them 9–12 months after treatment, and even lingering up to 24–48 months later. It points to a lasting shift in the immune environment after treatment, which is definitely a positive sign.
Okay, so there are signs treatment helps the immune system recalibrate. But even with all the focus on new drugs and immune responses, we shouldn't forget the current drugs, benzimidazole and nifurtimox, still pack a punch, right? Especially early on.
Or absolutely. Despite all the drawbacks we discussed, especially the side effects, when you look at the primary efficacy data, particularly for acute infections, more than 80% of patients who get active benzimidazole treatment achieve what's called sustained parasitological clearance, meaning the parasite is gone from their blood. Compare that to only about 3% in the placebo groups.
Wow, 80% versus 3%. That's dramatic.
It is. It really underscores that the drug does work powerfully against the parasite, which is exactly why researchers are putting so much effort into finding ways to make it safer or deliver it better, because it's still a vital weapon, especially early on.
Okay, let's pivot to some really groundbreaking studies. You mentioned one that caught my eye, an in vivo study. In actual living animals, not just cells in a dish, using that B. uncinella extract again.
Yes, precisely. They investigated the hydroalcoholic extract of B. uncinella and also a specific compound isolated from it called plus sanif-15 hydroxylab 7N17L. They tested this in a standard mouse model for Chagas disease, BALBC mice. Treatment started 7 days after the mice were infected.
And what did they find?
Really promising results. The treatment significantly reduced the number of parasites circulating in the blood. And perhaps even more impressively, it markedly increased the survival rates of the infected mice.
That's huge. Moving from a petri dish to showing actual survival benefit in an animal model, that's a critical step.
It absolutely is. It's essential validation for moving towards potential human trials down the line.
And besides finding totally new compounds, are researchers also getting clever with how they deliver the existing drugs?
Yes, that's another really important avenue. Improving treatment isn't always about finding a brand new molecule. Sometimes it's about making the ones we have work better or safer. So, for instance, people are exploring advanced formulations, nanostructures, things like cyclodextrin's benzimidazole complexes. The idea is to essentially package the benzimidazole molecule inside a tiny carrier, a nanostructure.
Like a tiny delivery truck for the drug.
Kind of, yeah. The hope is that this could lower the overall toxicity, Maybe target the delivery better, reduce exposure to healthy tissues. If you can make benznidazole safer and more tolerable, patients are more likely to complete the long treatment course. That addresses a major real-world problem.
Okay, so we've covered a lot of ground. Let's just quickly run through the main types of potential treatments we've touched on again. It's quite a diverse landscape.
It really is.
Yeah.
So we started with the established drugs, right? Benznidazole and nifurtimox. Effective, especially acutely, but with those significant tolerability and chronic phase efficacy issues. Then the targets within the parasite itself. Trypanothione reductase and squalene synthase, key enzymes researchers are trying to block. On the synthetic side, we have those organometallic shift bases and the triazole-based amides and sulfonamides, new chemical approaches. Then the plant-derived options, specifically compounds from bee uncinella like caffeic acid, pictolinaringen, hispidulin, and that mix of cryogenic acids, showing promise and low toxicity. And finally, the delivery innovations like those nanostructure formulations, the cyclodextrin complexes, aiming squarely at improving safety and tolerability.
That diversity brings up another question. Are scientists looking at combining these approaches? Synergy, sometimes hitting a problem from multiple angles is more effective, isn't it? Two compounds together, or maybe a drug plus a delivery system.
Absolutely. Synergy is a huge area of interest in drug development, and we saw examples right here in our sources. Remember that mixture of 3 cryogenic acids from bee uncinella, the 3,4, 3,5, and 4,5-decaffeoylquenic acids. The study highlighted that the mixture showed activity against T. cruzi, not necessarily just one isolated compound being the star. That strongly suggests these natural components might be working together synergistically. Nature often uses complex mixtures.
Like a team effort within the plant extract itself.
Exactly, a botanical team effort.
And thinking about combinations in a different way, those nanostructures you mentioned, like the cyclodextrins with benznidazole, that's essentially combining the drug with a delivery technology, right? The goal there is synergy too, isn't it? To get a better outcome, reduce toxicity, better tolerability than the drug alone could achieve.
Precisely. It's a combination approach focused on improving the drug's profile, rather than just its direct antiparasitic action. It's about making the treatment work better for the patient. Better adherence, fewer side effects, potentially leading to better long-term outcomes. That's a powerful form of synergy.
Wow. Okay, so we've really taken a deep dive into the intricate world of Chagas disease research today, from the parasite's biology to human trials and new tech.
We have. And it reinforces that Chagas, particularly those chronic heart and digestive forms, is still a major global health burden, and that the current mainstays, benznidazole and nifurtimox, while useful, have real drawbacks, especially for adults in the chronic phase, creating this urgent need for innovation.
But it's also clear the scientific community is pushing hard on multiple fronts. It's not just one track. We've seen targeting specific parasite weak spots like trypanothione reductase and squalene synthase, developing completely new synthetic molecules, systematically exploring plants like bee uncinella, finding active compounds, and even getting exciting results in animal models. That's really encouraging.
Definitely. And the focus isn't just on killing the parasite, it's also on the patient experience. Things like nanocarriers to make existing drugs less toxic, detailed human studies tracking how the immune system responds, even testing non-traditional approaches like selenium. It all points to this really determined global effort to find safer, more effective ways to manage this neglected disease. It's quite inspiring, actually.
It really is. So as we wrap up, thinking about this complex disease and this multi-pronged scientific assault against it, it really makes you wonder, doesn't it? Which of these different paths, the new synthetics, the natural products, the delivery systems, the immune modulation, or maybe some completely new combination we haven't even seen yet, which one will finally lead to that breakthrough? The breakthrough that could truly change the future for the millions of people living under the shadow of Chagas disease.